Hi, everyone. Welcome back. Welcome to your second half of our Human Anatomy course here in the pre-health program. We're going to begin the course looking at the cardiovascular system. And then in the third week, we're going to move into looking at how the lymphatic system is also tied in with the cardiovascular system. This week, we're going to focus on the heart, specifically the anatomy, as well as some of the physiological aspects of how those electrical signals are conducted throughout the heart muscle, which cause it to be able to contract. In the upcoming weeks, we're going to spend our time then looking at all the various arteries as well as veins that make up both our systemic circulation as well as pulmonary circulation. Now, the main purpose of the cardiovascular system is to be able to circulate blood throughout the body. And if you think back to our lessons on cellular respiration, specifically aerobic respiration, you'll remember that our cells require oxygen in order to further break down glucose through the Krebs cycle and then finally electron transport chain to produce ATP. The way that we get the oxygen that we need is by breathing it in through our respiratory system and then have that oxygen diffuse into the capillaries, which then connect directly to the pulmonary veins. Once that oxygen is in the blood, it circulates back to the heart, and then the heart then sends that oxygenated blood out to the rest of our body. So ultimately, we're going to be sending the oxygenated blood through the systemic arteries. And when the blood arrives at the cells that need that oxygen, the oxygen diffuses from the bloodstream into our cells. And then what is it that we produce as a byproduct of aerobic respiration? Right, our cells need the oxygen to be able to produce the ATP, but during pyruvate oxidation as well as the Krebs cycle, we produce carbon dioxide. And so that carbon dioxide can then move back into the bloodstream and be transported through the venous system in our deoxygenated blood all the way back to the right side of the heart. Then you can see how this connects here, whether it's coming from the upper part of the body or the lower part of the body. All of that deoxygenated blood empties into the right side of the heart, which then pumps it back out to the lungs. And think about this. What do we breathe out? Carbon dioxide. So we're spending our first 3 weeks focusing on the cardiovascular system, and then in weeks 4 and 5, we're going to move into looking at the respiratory system. And by the time we've completed all of that content before the midterm, you're going to have a full understanding of how it is that we go about bringing in that oxygen, circulating it throughout the body so that we can use it for aerobic respiration, and then taking the CO2 that we produce in our cells and circulating it back to the heart, out to the lungs, so we can ultimately breathe it out. Now, there are two main circulatory pathways that we're going to focus on, and those are the systemic circulatory routes and the pulmonary circulatory route. All right, also known as systemic circulation and pulmonary circulation. Now, systemic circulation covers all of the blood vessels that are going to be supplying all parts of our body with blood. Now, with systemic circulation, we're looking at the blood vessels, both arteries, veins, and capillaries, that are supplying the tissues of the body. Whereas with pulmonary circulation, we're looking specifically at the blood vessels that are taking deoxygenated blood towards the lungs and oxygenated blood away from the lungs. We're going to get into looking at all of the different vessels in a lot more detail in the coming weeks. But for now, if you understand, arteries are always going to be sending blood away from the heart, and the veins are always going to be sending blood back towards the heart. That's sufficient at this point. Now, the ways in which the arteries and the veins are connected continuously is through these capillary networks. So if you look here, you can see we have these systemic arteries, okay, circulating blood away from the heart, right? It's an artery. They connect then directly to these capillary networks. And the capillaries are the thinnest, smallest blood vessels in the body. Remember, they allow for oxygen and carbon dioxide to be able to diffuse in and out of the blood because they're made up of simple squamous epithelial tissue. Now, the way that the capillaries are connected to the arteries and veins is through these smaller blood vessels known as arterioles and venules. And so just generally speaking, the way that this works is that blood is going to circulate away from the heart through the arteries, and then it's going to move into some off ramps known as the arterioles. And the arterioles then are going to continue into the capillary network. And then from the capillaries are then going to join to form venules, which are like the on ramps, and then finally collect into major systemic veins throughout the body. All right, so this is just a general schematic at this point. We're going to get to spend a lot of time focusing on the specific names as well as locations of the different arteries and veins in the body. Now, the main functions of the cardiovascular system are transportation, regulation, as well as protection. Now, with transportation, we've already sort of talked about this in terms of transporting oxygen and carbon dioxide. When the blood is rich in oxygen, as it is here in the systemic arteries, we call that oxygenated blood. And when it's oxygen poor but carbon dioxide rich, as it is in the systemic veins, we call that deoxygenated blood. All right, and so one of the primary functions of our cardiovascular system is to be able to transport the oxygen to our tissues and then take the carbon dioxide byproduct, circulate it back to the heart so that it can go out to the lungs and we can breathe it out. Now, some other substances that we are capable of transporting throughout the body are various nutrients that we take in, wastes, as well as hormones. We've already looked at negative feedback mechanisms such as body temperature regulation, as well as blood pressure regulation, when we looked at how the sympathetic and parasympathetic nervous system are involved in helping to regulate blood pressure last semester. One of the ways in which our cardiovascular system can help to regulate body temperature is through the process of vasoconstriction or vasodilation. So these two terms, remember, are referring to whether we have a dilated vessel, okay, where we're opening or expanding it to allow for more blood flow into the capillaries to get rid of some of the excess heat, or we can vasoconstrict, right? And this comes back to looking at your hands. Say you go outside and you're cold. Why is the skin of our hands going to turn a little bit of a purple color or a bluish color? It's because our blood vessels are vasoconstricting to prevent as much heat loss as possible. Now, in the summer months, when it's a lot warmer or when you're working out and you're sweating, all of our blood vessels dilate to allow more blood flow into our tissues so that we can release a lot of that heat from our body. And lastly, we're going to spend 3 weeks looking at the immune system at the end of biology. And so we're going to get into looking at the protective mechanisms of our blood, such as the white blood cells, T and B lymphocytes that are all involved in providing us with immune protection. Now, this should look familiar to you. We went over all of the different types of muscle tissue last semester when we looked at tissues. So now we're going to be looking at the heart today. So what type of muscle tissue makes up the heart? Well, it's the cardiac muscle tissue. Remember, cardiac muscle tissue is striated, involuntary muscle tissue. The only place you find it in the heart. Now, one thing we had mentioned last semester was that the cardiac muscle cells contain a much higher amount of mitochondria than do other muscle cells in the body. And remember, mitochondria are involved in energy production. And so because the heart never gets a break, it's always contracting. Since it was formed, it's always contracting. You need a lot of ATP to be able to stimulate all of those individual muscle cells. And now a way that we can transmit the electrical signals smoothly throughout and make sure that those cardiac muscle cells are anchored to one another are those special features known as the intercalated discs. Remember, the intercalated discs are made up of two different types of junctions. We have the desmosomes that anchor the adjacent cardiac cells together. And then we also have the presence of gap junctions. And the gap junctions are there to allow for sodium to be able to rush in and out of the cells to be able to produce those action potentials. Now, to just review, we are looking at this individual here in anatomical position. So it's important for you to be able to know which side of the heart you're looking at. Are you looking at the anterior surface or are you looking at the posterior surface? We will spend most of our time focusing on the anatomy of the heart in anatomical position. Okay, so because we're looking at this individual in anatomical position, we know that the sides are flipped. We have the left side here and we have the right side over here. The heart is located within the thoracic cavity. Remember our superior ventral cavity. And the thoracic cavity, if you recall, is subdivided into 3 main subcavities. We have the lateral cavities that house the lungs, and then we have this central cavity that houses the heart as well as a lot of other key structures that pass through the midline of the thoracic cavity. And that's known as the mediastinum. Now, the heart is roughly the size of a closed fist, and about 2/3 of the mass lies to that left of the mid-sagittal plane, whereas 1/3 sits on the right side. Now with the heart, typically we expect the base to be on the inferior aspect and apex to be more superior. It's flipped here with the heart. So the apex of the heart is this portion right here that sits directly on top of the diaphragm. Remember, the diaphragm is the boundary that separates the thoracic cavity from the abdominal cavity. And conversely, the base of the heart is located at the superior aspect of the heart. The base of the heart is where you have all of the blood vessels connecting to the superior aspect. All right, so our major veins, the superior and inferior vena cava, you have the aorta, as well as the pulmonary trunk. And here we've included those serous membranes that surround all of these structures. We have, within the mediastinum, we have another cavity known as the pericardial cavity. And the pericardial cavity is bound by the pericardium, which is this outer serous membrane that covers the outside of the heart. Now, as with all serous membranes, the pericardium is a double-layered membrane, right? And between those two layers of the serous membrane, there is a space, and within that space, we produce serous fluid. Now, because this is the pericardium, we produce pericardial fluid. And remember, the purpose of that fluid is simply to provide decreased friction through lubrication to allow for ease of movement between these two opposing surfaces. So if you recall with the serous membranes, we always have one layer that's going to be connecting to the organ, and we have an outer layer that's going to be connecting to more superficial structures. And in that case there, the outer layer is the parietal pericardium, and the inner layer known as the visceral pericardium. Now, the heart itself is composed of three main layers of tissue. We're going to focus here, because it's the largest, on the walls of the ventricles, these larger chambers that you can see inferiorly here. The atria are also made up of these three layers. And these three layers are the epicardium, the myocardium, and the endocardium. All right, so let's go back. Let's take a look at our prefixes. Myo refers to muscle. So the myocardium is the name for the muscular layer of the heart. And you can see here in the ventricles, the myocardium is very thick. And the reason it is because of where the blood is having to be pumped from the ventricles. Okay, and it is always out of the heart, away into either the body or into the pulmonary circuit towards the lungs. All right, so the epicardium is located outside of the myocardium, and the endocardium located within. So if you were to cut open the heart and take a look at it here through this coronal or frontal section, you would see that the inner portion of the chambers would be all shiny and smooth. That shiny, smooth surface is the endocardium, and it's made up of connective tissue, and it's to allow for blood to be able to seamlessly flow into and out of the heart chambers. Blood does not like to sit still. When it does, it tends to clot. And so blood cells need to continue to move fluidly from one chamber to the other to then be ejected from the heart. Now, finally, that last layer, the epicardium is continuous here with the visceral layer of the pericardium. We can think of them as one in the same. So the three layers from most superficial to deep, we have the epicardium, myocardium, and endocardium. And then to put it all together, we have the parietal pericardium as the outer portion of the serous membrane. We have the epicardium, also known as the visceral pericardium. And between those two layers here, this white line, that is the pericardial cavity, pericardial space. This is where that pericardial fluid is going to be circulating and bathing these two opposing membranes to reduce friction when the heart's contracting. Deep to the epicardium, we have the myocardium, the muscular layer. And then lining the inner surface of the chambers, we have the endocardium. Now, as I mentioned before, we're going to be spending our time focusing primarily on the heart in anatomical position, but there are some structures that you can really only visualize appropriately if you turn the heart around and look at it from a posterior perspective. All right, so make a note here. We are looking at the heart in this image, in anatomical perspective, okay, so an anterior view. And here we're looking at the heart from a posterior perspective. Now, as mentioned previously, the apex of the heart is located at the inferior portion here. Remember, the apex lies directly on top of the diaphragm. Now, the base of the heart located at the superior aspect here is where you find all of those greater arteries and veins that connect the heart to the rest of the body. Now, as seen previously from an internal perspective, you saw that there were four different chambers, two upper chambers and two lower chambers. The upper chambers are known as atria, or a single one would be known as an atrium. The ventricles are the lower chambers. And the blood vessels that are going to then be supplying the myocardium with oxygenated blood, those are known as coronary arteries. So from an external perspective in anatomical position, we're looking at the right atrium, the left atrium, just a small portion of it, the right ventricle, and the left ventricle. And now if we turn the heart around, looking at it from a posterior perspective, we can see here that we are looking then at left is on left, right is on right, right? Because we're looking at this heart from a posterior view. So we have here in the upper chambers, we have the left atrium, we have the right atrium. Lower, we have the left ventricle and the right ventricle. Two features that we can visualize from an external perspective are known as the coronary sulcus and the interventricular sulcus. So if you think back to our lecture on the nervous system, a sulcus was a depression, right? When we looked at the brain, we had a gyrus, which was an elevation of the brain, and a sulcus, which was the depression. And so here we have these depressions on the outside of the heart that divide structures anatomically. Now, the coronary sulcus, think of it as the crown, all right? It wraps right around here and separates the upper chambers from the lower chambers. Okay, so the coronary sulcus is the external landmark separating the atria from the ventricles. Okay, it's really easy to visualize that right here. So all along here. So all of this area along here would be the coronary sulcus. Here you see a little bit here wrapped around, and you can't really visualize it too well on the left side there. Now you'll notice it's all yellow. What is that yellow tissue that you see surrounding the heart? Well, that would be your adipose tissue. So within the coronary sulcus, you have an abundance of adipose tissue, and that's what you're going to be using as a landmark to identify this structure. Okay, so the coronary sulcus separates the atria above from the ventricles below. Now the interventricular sulcus, let's break this down. Inter means between. Right? And ventricular is referring to the ventricles. So the sulcus that is between the ventricles, all right? So the interventricular sulcus, you can see both from a posterior as well as an anterior view. This is the depression that is identifying externally the separation between the right ventricle and the left ventricle. And then here we're looking from a posterior view. So the interventricular sulcus is separating the right ventricle from the left ventricle. Okay, so coronary sulcus separates atria from ventricles, and the interventricular sulcus separates the left from the right ventricles. And remember, these are external landmarks on the heart. Now, here we are looking at the heart again in anatomical perspective. We have taken what type of section? A coronal or frontal section, right? That's the one that cuts the face off. So sliced right off, the front anterior half of the heart has been removed, and now we can visualize the internal anatomy of the heart perfectly. Now, the heart is divided into left and right sides, right? So we have the upper chambers, the atria, you have the right and the left, and then you have the right and the left ventricles. Now, the atria are known as the receiving chambers because they are going to be receiving blood either from the body, if we're looking at the right atria, or from the lungs, if we're looking at the left atria. Okay, so they are known as receiving chambers. So all of that deoxygenated blood that's coming from the tissues of our body, it's all going to collect into two major blood vessels, the superior vena cava and the inferior vena cava. And all of that deoxygenated blood is just going to simply pour in to the right atrium. All right, so the right atrium receives deoxygenated blood from the body. The left atrium, remember, once all of that deoxygenated blood eventually circulates out of the pulmonary arteries towards the lungs, it's going to pick up oxygen and give off the carbon dioxide. So now that blood is oxygenated, and that oxygenated blood is going to travel back to the heart, to the left side, and be received by the left atrium. Ultimately, the blood that's in either atrium is going to be transferred from that chamber to the ventricle immediately inferior to it. All right, so from the right atrium, blood is going to pass into the right ventricle. And from the left atrium, blood is going to pass into the left ventricle. So as we mentioned before, we have deoxygenated blood in the right atrium. We are going to have deoxygenated blood in the right ventricle. And here we've received oxygenated blood in the left atrium. That oxygenated blood will be transferred into the left ventricle. From the ventricles, they are going to contract and ultimately discharge blood from the heart. And this is why the myocardium of the ventricles is so thick, because the blood from the right ventricle is going to be discharged out and has to be transferred over towards the lungs. And then all of the blood in the left ventricle is ultimately going to be sent out to the rest of the body. And so you'll notice, comparing the right to the left ventricle, if you take a look at the myocardium, you can see here that the myocardium of the right ventricle is significantly smaller than the myocardium that makes up the left ventricle. And it's ultimately because the left ventricle is going to have to really work to contract to send all of that blood out from the heart to the rest of the body. All right, so the atria are the receiving chambers, and the ventricles are the discharging chambers. And as you can see now, the right side of the heart contains deoxygenated blood, and the left side contains oxygenated blood. The superior and inferior vena cava are both veins, and they are responsible for draining structures located above the heart or below the heart. All right, so in tutorial, I'm going to be able to show you this a little bit better, but if you all go into sort of a Superman position, abduct your arms so that they're horizontal to your clavicle, all structures above your heart in that position, including your upper extremities, they're all going to drain their deoxygenated blood back through the superior vena cava into the right atrium. Now, in that same position, all structures located below the heart are going to be draining blood through the inferior vena cava back to the right atrium. Two other structures that you can see here are known as the fossa ovalis and the coronary sinus. Now, the coronary sinus is responsible for emptying all of the blood coming from coronary circulation. So previously we looked at the coronary arteries that are going to be supplying the heart muscle, the myocardium, with oxygenated blood. Well, once the heart muscle, the cardiac muscle, has used up that oxygen, it's going to produce CO2. And that CO2-rich blood, the deoxygenated blood, is going to drain back through specific veins known as cardiac veins. And all of those cardiac veins are going to collect and empty into the coronary sinus, which again empties into the right atrium. So we have a way of being able to empty all of the deoxygenated blood from the body, as well as from coronary circulation, back into the right atria. And from there, we can circulate through into the right ventricle, out to the lungs to get rid of that CO2 and pick up that oxygen. Now, this other structure here known as the fossa ovalis, the fossa ovalis is a remnant structure. We'll get to take a look briefly at circulation of a neonate, so of a fetus. And within a fetus, there are a number of bypasses because they don't need their lungs for oxygen, right? And one of the bypasses that they have is a hole that connects the right atrium to the left atrium. And once that baby is born and they start crying, taking that first breath, the increased pressure closes off the hole connecting the right atrium to the left atrium. And the remnant structure is known as the fossa ovalis. Some key identifying structures that then are going to empty into the left atrium, and we can see this here from anatomical position, as well as here looking at posterior view. You can see the pulmonary veins. So pulmonary refers to the lungs, okay? So we know this is somehow connected to the lungs. Veins, remember, are always going to be taking blood and sending it back to the heart. So if the blood's coming from the lungs back to the heart, you know it must be oxygenated blood, right? So the pulmonary veins deliver that oxygenated blood into the left atrium. You can see here as well from the left lung, you have the pulmonary veins that are taking that oxygenated blood to the left atrium. Now, once that blood has been received in either the right atrium or the left atrium, depending on where it's coming from, those atria are going to contract. And when they contract, they are going to eject blood from the atria into the ventricles. So from the right atrium into the right ventricle, or from the left atrium into the left ventricle. Remember, we're looking at the right side of the heart, so you're looking at deoxygenated blood. And here with the left atrium, we're looking at left side, so you know it's oxygenated blood. Now, once that blood is in either right or left ventricles, the ventricles are going to be responsible for contracting and then ejecting that blood out of the heart. So if it's deoxygenated blood, it needs to become oxygenated. So where do we get the oxygen from? Our pulmonary circuit. And so ultimately, the right ventricle is going to send the deoxygenated blood out of the heart through a structure known as the pulmonary trunk. And the pulmonary trunk then connects directly to the pulmonary arteries. So remember, arteries, always away. Veins, always back towards. Now, once that deoxygenated blood is sent out to the lungs, we get rid of the CO2, we pick up O2, oxygen, and that oxygenated blood now is going to circulate back through the pulmonary veins and empty into the left atrium. That left atrium contracts and sends that oxygenated blood down into the left ventricle. The left ventricle is going to contract, and remember, it has that huge myocardium because the blood in the left ventricle is destined to be sent throughout the rest of the body through this large blood vessel known as the aorta. And here we're looking at an oblique cross-section that's been taken to help us to visualize the muscle that makes up the walls of the ventricles. And so you can see here the wall of the right ventricle much smaller than the wall of the left. And that's simply because the right ventricle just has to send the blood out to the lungs, pretty close, whereas the left ventricle has to send all of that oxygenated blood out of the heart to the rest of your body. Okay, and as a quick recap, let's quickly review the external anatomy of the heart together. So let's run through this in terms of sequence. Here you have the apex of the heart looking at anatomical perspective here, or the apex looking in a posterior perspective. The apex, remember, sits directly on top of the diaphragm and is located at the inferior portion of the heart. Moving superiorly, we are now looking at the base of the heart, and the base of the heart contains all of the major blood vessels. You have the superior and inferior vena cava, you have the pulmonary trunk, as well as the aorta. Okay, and from a posterior perspective, you can also see the base of the heart where you would have all of these blood vessels connecting to. The receiving chambers located superiorly are known as the atrium. So in anatomical position, we are looking at the right atrium and the left atrium. Looking in posterior, we are looking at the left atrium and the right atrium. The inferior discharging chambers of the heart are known as the ventricles. Here we have the right ventricle, and then past the interventricular sulcus, we have the left ventricle. And you can see most of the left ventricle is visible here from the posterior perspective, and here you can see a little bit of the right ventricle from the posterior perspective. And last but not least, we can also visualize the coronary sulcus, which divides the upper receiving chambers, the atria, from the lower discharging chambers known as the ventricles. Now, throughout the heart, we have a number of one-way valves. And the reason that we have these one-way valves is to prevent blood from being able to flow in a retrograde fashion. Now, what does that mean? It means we want to keep blood moving in a forward direction. We don't want any backflow. And when you have an issue with any one of these valves, you end up with what's known as an incompetent valve. And it can then allow blood to move in a retrograde fashion. What's the issue with that, you might ask? Well, you're going to need to then have your heart working a lot harder to be able to pump the same amount of blood through the rest of your body to supply the cells with the oxygen that they need. So first off, we're going to take a look at the atrioventricular valves, also known as the AV valves. And you can see here the AV valves, as their name describes, are separating the atria from the ventricles. And so we have a right atrioventricular valve, and we have a left atrioventricular valve. Now, these atrioventricular valves also go by different names, and that is based on anatomically how many cusps they have. And so here on the right side, the right atrioventricular valve is also known as the tricuspid valve. And the left atrioventricular valve is also known as the bicuspid. Okay, so the right AV, because there are 3 cusps, it's known as tricuspid, and the left AV is known as bicuspid because there are only 2. Okay, so 2 portions make up the bicuspid and 3 cusps make up the tricuspid. Now, just to make things even more fun, the left atrioventricular valve, also known as the bicuspid valve, also goes by a 3rd alias known as the mitral valve. Okay, so make sure you put that to memory. We have three different ways of describing this one valve. Okay, so it's the left atrioventricular, also known as the bicuspid, also known as the mitral. Now, both of these atrioventricular valves are connected to the endocardium in a very unique way. And that is through these connective tissue-like tendons known as the chordae tendineae. And these chordae tendineae connect directly to these extensions within the ventricle known as the papillary muscles. So remember, the muscular layer here, the myocardium, is going to extend into the ventricle as these structures known as the papillary muscles. And then all of that is going to be covered in that endocardium, right? That inner thin connective tissue layer. So ultimately, the chordae tendineae are connecting the atrioventricular valve directly to the papillary muscles. And this allows for control of the valves. So when the ventricles contract, which way do we want blood to flow? Out of the ventricle, right? Because the ventricle is the discharging chamber. So when the ventricle contracts, that's going to cause the right atrioventricular and the left atrioventricular valves to slam shut. And that way, blood is then forced to leave the heart into its respective vessels. Now, there are two other types of valves, and these are the valves that blood is going to pass through when it is leaving the ventricles and exiting the heart. Right? So they are known as semilunar valves. They work sort of as a one-way door. So when the ventricles contract, blood is allowed and forced to move through them in one direction. But when the ventricle relaxes and blood that's left here in the trunk might want to then move backwards, those semilunar valves are going to slam shut, similar here in the aorta. So these semilunar valves, when healthy, allow for blood to flow in one direction, but will slam shut, preventing any retrograde flow back into the ventricles. Okay, and they are named based on the vessel that the blood is going to be flowing into. So here from the right ventricle, blood is going to be sent through the pulmonary semilunar valve, right? Because where's its destination? The pulmonary trunk. And here from the left ventricle, blood is going to be ejected through the aortic semilunar valve, because it is then destined to pass through the aorta as it sends off to the rest of the body. So this gives you a different visual here. When the ventricles are contracting, the semilunar valves are going to be open because the blood is being forced out of them. But then when the ventricles relax, the blood is going to want to then flow backwards, back down, just due to gravitational forces. But those semilunar valves are going to close and prevent the blood from moving any further. This is what allows us to move that blood in one direction, both the presence of the atrioventricular valves and the semilunar valves. So if we take another look here at an oblique cross section through the heart, you can see all of these valves visualized. And so here, comparing the right and the left AV valve, you have a real appreciation here for the three cusps that make up that tricuspid valve versus the two that make up the bicuspid or mitral valve. And then you can visualize here for the aortic semilunar valve as well as the pulmonary semilunar valve. So now we're going to run through blood flow through the heart. As I mentioned before, if you divide the heart right down the midline, the right side of the heart contains deoxygenated blood, and the left side of the heart always contains oxygenated blood. The deoxygenated blood, remember, has been created from the carbon dioxide entering into our bloodstream at the level of the tissues. And so all of those veins throughout the body are going to transport that deoxygenated blood that has a lot of carbon dioxide in it back to the right atrium. All right, and so how does it do this? Well, all tissues that are above the heart are going to drain back through the superior vena cava into the right atrium, and all structures located below the heart are going to drain back into the right atrium through the inferior vena cava. All of the systemic deoxygenated blood drains back into the right atrium through the superior and inferior vena cava. Then once the deoxygenated blood is in the right atrium will contract and send that deoxygenated blood through the tricuspid, or right atrioventricular valve, into the right ventricle. Then when the right ventricle contracts, that tricuspid valve is going to slam shut, and all of that deoxygenated blood is going to be forced through the pulmonary semilunar valve into the pulmonary trunk. From there, splits off to the left and the right sides through the pulmonary arteries flowing away from the heart, where it's then going to be transported to the lungs. Now, once that carbon dioxide is given off in the lungs, we breathe it out, and then we breathe in new oxygen. Well, the oxygen then that's in our lungs is going to diffuse into the bloodstream, and that oxygenated blood is going to pass back towards the heart through the pulmonary veins. The oxygenated blood then transports into the left atrium. That left atrium contracts and sends that oxygenated blood through the bicuspid, mitral, or left atrioventricular valve into the left ventricle. That left ventricle will then contract and send that oxygenated blood out of the heart, passing through the aortic semilunar valve as the blood then passes directly into the aorta. And the oxygenated blood is then sent throughout the rest of the body through our systemic circulatory route. All right, so with the right side of the heart, we're looking at deoxygenated blood. It's coming from the body and it needs to be sent out to the lungs. And then the left side of the heart contains oxygenated blood, and the oxygenated blood has come from the lungs, and that is to be sent out to the rest of the body through the aorta. Now, the way that we regulate the contraction of this cardiac muscle is through autonomic nervous innervation. So we've covered this. Remember the parasympathetic, the sympathetic nervous system? Well, the heart receives dual innervation, right? It receives innervation from both the sympathetic and parasympathetic. So when you're stressed out, the sympathetic division is going to be stimulating the heart and heart rate is going to increase. When the parasympathetic stimulates the heart rate will decrease. And that is how our heart rate is regulated. Now, one single heartbeat is known as a cardiac cycle. We're going to go through all of the different phases here from an electrical perspective as well as a functional perspective in just a minute. But every heartbeat, that lub-dub that you would hear if you listen to your heartbeat with a stethoscope, that is one cardiac cycle. But typically the average pulse rate or average number of cardiac cycles per minute are 72 beats per minute. That's a good number to remember. And we're going to go over some ranges because a normal range is about 60 to 100 beats per minute. 72 beats per minute is just the average heart rate. Now, the average duration of one cardiac cycle is about 0.8 seconds. So if you do the math, you have 0.8 seconds, you have 60 seconds in a minute, that works out to your 72 beats per minute. All right, so if your heart rate's higher, your average duration is going to be a lot lower. And if your heart rate is lower than 72 beats per minute, you're going to have much longer duration of your cardiac cycles. Now, as we've looked at blood flow through the heart, we've looked at it sort of from one side out to the lungs, and then looking at the other side and then out to the body, right? We've sort of separated it. But one thing that's important to understand is that the atria are going to be contracting on both sides of the heart at the same time. And that state of contraction, whether it's the atria or the ventricles, is known as systole. Okay? Systole is the contractile phase, whereas diastole follows systole. Diastole is the relaxation phase. So once your atria have contracted, they've sent that blood into the ventricles. The atria are then going to relax. And when they do, the ventricles are going to contract, and the ventricles will then be in ventricular systole. So as we look at the cardiac cycle in more detail, you're going to see that the atria contract together during atrial systole while the ventricles are in diastole. And then when the ventricles are in systole, when they're contracting, the atria will be in diastole. They will be relaxed. Okay, so when one group of chambers is in systole, the other is in diastole, and then vice versa. And so here you can see both instances identifying contraction. So we're looking at systole in both of these images. So in this image here, we're looking at during atrial contraction, that would be atrial systole. So blood is being ejected from the atria into the ventricles. When the atria are in systole, the ventricles are in diastole, right? The ventricles have to be relaxed because they are going to be just filling up with all of that blood that's coming from the atria into the ventricles. Now, when the ventricles contract, remember these atrioventricular valves are going to slam shut. And so the ventricles contract, they are in systole. Well, now the atria are in diastole again, because as these valves are shut, both atria are going to be filling up with blood again for the next cardiac cycle. Okay, so systole is contractile phase, diastole, the relaxation phase. And ultimately, what controls the rate at which the atria and the ventricles are going to be contracting is all regulated by the conductive system of the heart. We have very specialized neurological structures located within the heart, primarily here within the right atria, and they are responsible for setting the pace and then also creating a bit of that delay between atria and ventricular systole. Because remember, when you have any kind of nerve impulse, it travels at lightning speed. So if we didn't have a way to be able to slow down conduction from the atria to the ventricles, the whole heart would just contract all at once. And so we have a node known as the AV node that's responsible for sort of delaying the transmission of that subsequent impulse to depolarize the ventricles, causing them to contract. All right, so the conductive system is made up of a number of different structures. First off, we have the sinoatrial node, also known as the SA node. And the SA node is also known as the pacemaker of the heart. It is the neurological tissue that is going to be depolarizing at a consistent rate to be able to depolarize the atria and subsequently the ventricles. And what happens when you depolarize? You're going to be causing contraction to occur. The sinoatrial node is located here, the upper portion of the right atrium, near the opening of the superior vena cava. So this SA node is going to be dictating your heart rate. Right? And so if the sympathetic nervous system is going to be stimulating the SA node, well, then the SA node is going to increase the frequency of nerve signals and going to increase the frequency of contractions. Whereas if the parasympathetic nervous system is going to be innervating and affecting the SA node, that is going to be causing the SA node to reduce its rate. Okay, so every signal sent out from the SA node causes one cardiac cycle. In biology, we're going to get to focus on the cardiovascular system in more detail. We're going to get to look at pacemaker potentials, a different type of nerve impulse that is created here within the heart tissue itself. So the SA node is going to fire off action potentials and then depolarize both the right atria as well as the left atria. All right, there is nervous tissue that is going to be extending into that left atria. So when the SA node depolarizes, essentially both right and left atria will depolarize. And when they depolarize, the atria will then subsequently contract. Now, from the SA node, those electrical impulses are ultimately going to arrive at this other structure known as the atrioventricular node. Okay? And the atrioventricular node is also located in the right atrium, but instead it is located closer to the lower part of the interatrial septum that separates the right from the left atria. Now, the main goal here of the atrioventricular node is to cause a slight delay in transmission throughout the rest of the conductive system. Because remember, once you depolarize the atria will contract. Well, when the atria are contracting, the ventricles need to be relaxed. If you depolarize the whole heart all at once, everything's going to contract and blood is not going to move anywhere. So to be able to have the heart operate effectively, we need to be able to have the atria contract first, which is going to send the blood into the ventricles, which will then send the blood into the ventricles, and then through a slight delay, have then the ventricles become depolarized, which will then allow them to contract, while the atria will then relax. So after this slight delay caused here at the AV node, the signal is then sent along to the bundle of His. And the bundle of His is another neurological structure that is named after the individual that first initially described it. Now, from the bundle of His, we have left and right AV bundles, okay, which are also known as bundle branches. And these bundle branches are going to be transmitting that electrical signal down the interventricular septum towards the apex of the heart. Once here at the apex of the heart, those bundle branches then spread out and innervate the entire ventricular myocardium in the form of structures known as Purkinje fibers. All right, so through the interventricular septum, we have left and right bundle branches. And then from the apex of the heart, and you only see one branch here, but remember, this is a 3D structure. You're going to have Purkinje fibers surrounding the entire myocardium of the ventricles. And those Purkinje fibers are going to cause then a very nice, smooth depolarization of the ventricles, which will lead to ventricular contraction. So to recap, The conductive system of the heart is made up of the SA node, the AV node, the bundle of His, left and right bundle branches, and the Purkinje fibers. The SA node, which is the sinoatrial node, is also known as the pacemaker. And that's because it sets the pace, the rhythm at which your heart is going to contract. When the SA node depolarizes, it sends those electrical signals through the right atria, but also through the left atria. When the atria depolarize, they will subsequently contract. When that signal then arrives at the AV node has a slight delay before sending it further to depolarize the ventricular tissues. And when the ventricles depolarize, they will then also subsequently contract. And remember, the average cardiac cycle is about 0.8 seconds. So from the SA node firing to the depolarization of both atria and ventricles and subsequently contraction and relaxation of both, that takes on average about 0.8 seconds. So you can hopefully appreciate that when you're stressing your heart and your heart rate is elevated, your electrical conductive system having to operate seamlessly to be able to coordinate all of the contraction to ensure that the heart is going to be pumping the blood through it as efficiently as possible. Now, a way in which we can visualize the depolarization and repolarization events of this electrical conductive system is through the ECGs. So an ECG, I'm sure you've seen these all on TV somewhere. I know we talk about Grey's Anatomy a lot. For sure on Grey's Anatomy, we've seen these. These are those little blips you see, okay, that are monitoring heart rate. The ECG can tell us a lot about what's happening with the electrical conductive system of the heart. And first off, what you see here are three separate waves. We have the P wave, followed by the QRS complex, followed by the T wave. Now, the P wave is representative of the depolarization that's taking place within the atria. So when the SA node fires off an action potential and depolarizes the right and the left atria, we visualize that as this P wave. Then what happens after the atria are depolarized? Well, they will subsequently contract. Where does that signal end up? At the AV node, right? The atrioventricular node. And then that signal will then be sent through bundle of His to the bundle of branches to the Purkinje fibers. And ultimately, that whole loop is meant to depolarize the ventricles. And so that second depolarization we see here, which is huge, is known as the QRS complex. And this is representative of the depolarization of the ventricles, both right and left. So remember, we talked about before the left myocardium versus the right myocardium, significantly thicker because that blood has to be sent to the rest of the body. But if you compare even the myocardium of the atria versus the ventricles, it's very small compared to the ventricles because you only have to send the blood from the atria inferiorly into those ventricles. So not a lot of muscle tissue required there to do that job. If you look here then at the ECG, you can see that the depolarization that takes place within the ventricles is huge compared to that takes place within the atria. And that's simply because we are recruiting far more muscle tissue to be able to contract effectively, to be able to move that blood either from the right ventricle into the pulmonary circuit or from the left ventricle to the rest of our body. Now, what happens after ventricular polarization? Well, the ventricles are going to contract immediately after, and then they are going to eventually relax. And so one thing that you're seeing here in this T wave is the repolarization that takes place within the ventricles following the depolarization. And that repolarization is indicative of the relaxation that's going to be taking place after ventricular systole. You might be asking yourself, though, okay, but if we have depolarization and repolarization events taking place, where's the repolarization of the atria? Well, it's somewhere hidden in this QRS complex. The reason we don't see that atrial repolarization is because it happens while the ventricles are being depolarized. And so the wave we would see from the atrial repolarization is actually being masked by the very large depolarization we're seeing as the QRS complex. So to recap, we have the P wave, which represents the depolarization of the atria. Atria will contract following that. Then we have the QRS complex, which identifies the depolarization of the ventricles. What also happens here? Repolarization of the atria, but we're not seeing it as a separate wave because it is masked by the QRS complex. And then once the ventricles have depolarized, they will contract, and then they will repolarize, which will lead to them relaxing. And as we mentioned previously, the rate and rhythm at which your heart is going to contract, it's the SA node that's actually causing that signal, but it's the sympathetic and parasympathetic nerve fibers that are ultimately going to cause the SA node to increase its rate of firing or decrease its rate of firing. And the way that the postganglionic nerve fibers do this, the sympathetic nerve fibers are going to release norepinephrine. Now, with the sympathetic nerve fibers, they are going to innervate the entire heart muscle as well as the nodes themselves. And so when norepinephrine is released, it's going to be dumped on the entire heart itself. In contrast, the parasympathetic nerve fibers are only going to innervate this SA node, the sinoatrial node, and the atrioventricular nodes. And that is the way in which they're going to help to decrease heart rate. Okay, so remember, with sympathetic, it tends to always be over-encompassing, right? It's just a whole body experience. So the way that the sympathetic nerve fibers are going to achieve that result of increasing your heart rate is not only innervating the nodes to increase regulation, but also the muscle itself. Okay, so norepinephrine is going to have that impact of increasing heart rate, whereas acetylcholine will have the impact of helping to decrease the heart rate. Now here we can put those two pieces together, looking at the electrical conductive system here of the heart, as well as pairing that up with the ECG. Okay, so there are different measurements that can be taken between the P wave and QRS and between the QRS and the T wave, known as intervals, but do not need to worry about those for our purposes. As long as you understand what is happening at each of those major waves. So you have the P wave, the QRS complex, and the T wave. Now, arrhythmias, which are irregularities in heart rhythm, anytime you have the SA node disrupted in some way, which could be due to any type of cardiovascular pathology, coronary artery disease, hypertension, myocardial infarction, a heart attack. Remember, hypothyroidism, the thyroid hormone regulates your metabolism, and when you have an elevated level of thyroid in your body, that acts to increase your overall metabolic rate. Well, one of the ways in which that does that is by impacting the heart rate. And so heart rate for somebody with hyperthyroidism will tend to be elevated. Defective heart valves mean the blood is going to flow retrograde fashion. The heart's going to have to work overtime. So signals and feedback are going to be all confused. And then also stress and drugs and other chemicals can also cause other irregularities. You do not need to memorize any of these. I just brought them here from my other text to give you some ideas of what's happening here. And just hopefully you can appreciate they don't all look like that typical ECG that you would expect to see. So with complete heart block, you have a complete dissociation of the way in which the electrical signals are transmitted through the heart. So you have a dissociation of the P waves, the QRS complexes. With bradycardia and tachycardia, these refer to lower or elevated heart rates. And we'll get into these terms a little bit later, I think, too, next week. But brady means slow, tachy means fast. So remember, normal heart rate is between 60 to 100 beats per minute. You can see here with bradycardia, the cardiac cycles much longer between each one. And then with tachycardia, much shorter cardiac cycles. Now, one way we can measure the overall effectiveness of the heart is to measure the stroke volume, which is how much blood the heart is ejecting every beat, multiplied by your heart rate, how many beats per minute. That can give you your cardiac output. And the cardiac output is a measure of how much blood the heart is pumping every minute. Now, on average, and this is a good number to remember, the average heart is going to be ejecting about 70 milliliters of blood every beat. So if you multiply that by your ideal heart rate of 72 beats per minute, that is going to give you an overall cardiac output of 4,900 milliliters, close to almost 5 liters, right? 4.9 liters. Well, it just so happens that the average total amount of blood in the human body is about 5 liters of blood. So every single minute, you're moving all of the blood located in your cardiovascular system throughout the body. And so you can appreciate if someone were to go into cardiac arrest where their heart is no longer pumping, and we're used to having all of our blood circulating throughout our bodies 5 liters a minute, there is going to be a significant deficit in such a short period of time. Now, with our heart rate, as we've mentioned, that can be influenced by either the stress response, the sympathetic, or rest and digest, the parasympathetic response, right? So with the sympathetic flight or fight response, we're going to see an increase in heart rate, whereas with the parasympathetic rest and digest, we are going to see a decrease in heart rate. All of the control of the heart originates in the medulla oblongata within the cardiovascular center, right? And then from there, sympathetic and parasympathetic impulses are going to be specifically regulating the heart as to either increase for sympathetic or decrease, which is parasympathetic. One way in which we control heart rate is through this autonomic response received from baroreceptors. Now remember, think barometer, baroreceptors are detecting pressure changes. And we have a way of being able to slightly alter our blood pressure simply by either increasing or decreasing our heart rate. You don't need to worry about specifics here of the feedback loop. The main thing is to understand we have these receptors located in the aortic arch, as well as in the carotid arteries. Carotids are the main arteries that feed our head and our brains with oxygenated blood. So we have these baroreceptors located in these structures. This is where pressure is highest, right? Because it's coming from the left ventricle. Remember, left ventricle, very thick myocardium. So the blood pressure located in the aorta and the carotid arteries is going to be the highest outside of the heart. So we have these baroreceptors to detect pressure changes. If our blood pressure is gone up slightly, we have a way of being able to send signals to the medulla oblongata from those baroreceptors, which will then send parasympathetic signals, causing the overall heart rate to slow down. Vice versa, if we have a reduction in blood pressure, we need to increase our heart rate to bring that blood pressure up. And so a reduction in blood pressure would be received by the cardiovascular center in the medulla. And then the medulla would communicate directly with the sympathetic nervous system, causing the heart rate to increase. All right, so the change in blood pressure being detected by the baroreceptors sent to the cardiovascular center in the medulla, and then is going to be regulated by either increasing or decreasing heart rate. Okay, and it's always an inverse response. If your blood pressure is going up, you want to decrease your heart rate. And if your blood pressure is going down, you want to increase your heart rate. Okay, so that brings us to the end of our lecture for this week. So if you have any questions in the meantime, please feel free to reach out. Otherwise, I look forward to seeing you all back in tutorial this week. Take care.